Okay guys, we're going for a trip.
A few moments later.
What happened?
Wait, why the car stop?
I don't know. I think the car broke down.
OMG, where are we now?
There's no signal here.
So what are we going to do now?
Do we have food to eat?
6.5 hours later.
There are no cars or people at all.
I'm so hungry.
So, what will possibly happen to them? Don't worry, our body has an amazing survival mechanism called starvation mode. Starvation mode is the state your body enters when it doesn't get enough food or energy for a long time. During this time, your body adjusts how it uses energy to make sure vital organs like your brain, heart and lungs can keep working. It does this by use stored energy from 3 types of metabolism, which are carbohydrates, proteins and lipids.
The first one is metabolism of carbohydrates during starvation. During the initial phase of starvation, blood glucose levels drop, leading to a decrease in insulin secretion. In response, the pancreas releases glucagon, which inhibits glycogen synthesis in the liver and stimulates glycogenolysis. The glucose produced is released into the bloodstream to provide energy for cells, particularly glucose-dependent tissues like the brain and red blood cells. This process is regulated by glucagon and epinephrine. Glycogen stores in the liver and muscles are depleted within approximately 24 hours. Once glycogen stores are depleted, the body relies on gluconeogenesis to maintain blood glucose levels. Glucose is synthesized primarily in the liver from non-carbohydrate precursors such as lactate, glycerol, and amino acids. This process ensures a continuous supply of glucose for critical tissues like the brain and red blood cells, which still require glucose. As starvation progresses, the body reduces glucose utilization in most tissues to preserve it for glucose-dependent organs. To meet energy demands, the body increasingly relies on fatty acid oxidation for energy production and ketogenesis to produce ketone bodies as an alternative energy source for the brain and other tissues. For hormonal regulation, glucagon levels rise during starvation to stimulate gluconeogenesis and glycogenolysis, and insulin levels decrease to limit glucose uptake and storage in non-essential tissues.
What will happen to the lipid during starvation? For the first few hours to days, body will use glycogen stores through glycogenolysis and begin gluconeogenesis. But after 2-3 days, glycogen stores will become depleted and prompt a shift to lipid metabolism. Triglyceride will break down into fatty acids and glycerol. In the liver, fatty acid will be converted into ketone body through ketogenesis. This ketone body will serve as energy source for tissues and brain, while glycerol will undergo gluconeogenesis to produce glucose. Hormones such as glucagon and cortisol will promote lipolytic activity, which accelerate lipolysis and suppress lipogenesis. They will allow more efficient use of stored fat.
Now, let's talk about the metabolisms of protein during starvation. During starvation, proteins undergo gluconeogenesis. Gluconeogenesis is the production of glucose from non-sugar precursor. Before gluconeogenesis, there are 2 processes occur. First, protein degradation and amino acid transport. Muscle protein broken down during starvation to produce alanine and glutamine. These amino acids are then transported to the liver for gluconeogenesis. Second is showing deamination and transamination. Alanine undergoes transamination with the help of alanine aminotransferase to form pyruvate and ready for gluconeogenesis. Glutamine undergoes diamination via glutaminase to form glutamate, which in turn undergoes transamination to form alpha-ketoglutarate. The conversion helps provide additional metabolic intermediate for gluconeogenesis. Pyruvate is converted into oxaloacetate by the enzyme pyruvate carboxylase. This step requires ATP. Oxaloacetate is then converted to phosphoenolpyruvate by phosphoenolpyruvate carbokinase. Fructose 1,6-bisphosphate is converted to fructose 6-phosphate by the enzyme fructose 1,6-bisphosphatase. Fructose 6-phosphate is isomerized to glucose 6-phosphate by the enzyme phosphoglucose isomerase. Finally, glucose 6-phosphate is dephosphorylated by glucose 6-phosphatase to produce free glucose, which is released into the bloodstream to help maintain glucose homeostasis.
This is the energy yield comparison between carbohydrate, proteins, and lipid. During starvation, carbohydrate is our main source energy because they can quickly metabolize to produce 4 kilocalories per gram. However, the glycogen that stores in our liver and muscles are limited, so they can be depleted within 24 to 48 hours of starvation. As for proteins, they yield to 4 kilocalories per gram. So during prolonged starvation, when the glycogen that stores in our liver and muscles are being depleted fully, proteins will be broken down into form of amino acid. This is the important element for the gluconeogenesis process and production of energy. Meanwhile, lipid will produce 9 kilocalories per gram, making them the main energy source during prolonged starvation. Fat that stores are mobilized as triglycerides, so they will be broken down into form of glycerol, which used for gluconeogenesis, and fatty acid for the beta oxidation to generate ATP. That's why lipid is known as the main energy source during starvation.
Our bodies have incredible adaptive mechanism to cope with prolonged starvation. First, the body will use stored glycogen in liver and muscle that it stores for quick energy. When we stop eating, our bodies will use this first to keep our energy level stable. Second, when the stored sugar runs out, our body will start break down lipids into energy to keep us going. Third, our bodies will usually break down a little protein in muscles for energy first. But as starvation continues, the body will try to save muscles because they are very important for survival. Next, our brains normally use sugar for energy. But in starvation, the body makes ketone from the lipids as the main energy for the brain, so it doesn't need as much sugar. To save energy, our body will reduce how much it burns. This means you will feel tired and cold, and your heart and breathing slow down due to your metabolism slowing down. Lastly, our body cell will break down all or damaged body parts and use them for energy. It's like our bodies are recycling to survive.
To sum up, during starvation, within 24 to 48 hours, carbohydrates are known as the primary source of energy, while lipids are known as the main energy source for prolonged starvation. Meanwhile, proteins are being served as the last energy source, especially in an extreme starvation. So guys, please don't starve yourself, okay? Because it is very dangerous to our body.